chapter 9, now we're going to do genetics. The introduction to genetics with microbes. All the fun stuff now we've been talking about. So genetics is basically the study of heredity, how things are passed down from generation to generation of any species. Okay, it's a transmission of biological traits from your parent to your offspring. Okay, you get different expressions of those traits, and there's variations of those traits. We'll see as we go along. There's mutations that occur that could be good or that could be favorable. Okay, so the expression of those traits can change. You have the structure and function of genetic material, how DNA works, how basically those little bases, the sugar phosphate backbone with those adenine, thymine, guanine, cytosine bases, determine what proteins are made in the body. And then how this material can change. Okay, so these are the functions of a genome. Genome is the total amount of your genetic material. In this case, DNA. This is Enterobacillus vermiculus. This is a comparasitic worm. And this is what it looks like on the cellular level. You have your cell wall, your cell membrane, your nucleus, and then inside the nucleus, you have chromosomes, which are your DNA. And you take the chromosomes, enlarge that, enlarge the chromosome. You see that the chromosome is just a double strand of DNA that is packed together very tightly. It gets tightly wound so cells can replicate and duplicate. Okay. So the genome, when we talk about a genome, like the human genome, we're talking about the total genetic material, which is DNA of the entire cell. Most of the humans on eukaryotes exist as chromosomes. Okay, some up here in non-chromosomal sites, like mitochondria. We have mitochondrial DNA. Okay, we have chloroplast, and plasmids also contain DNA. Mitochondrial DNA has made a big thing on all the new CSI shows. That's where we get mitochondrial DNA because we can get that in greater amounts than regular DNA, like in the blood, things like that, that are left behind. You can isolate the mitochondrial DNA a lot easier. The genome of cells is DNA. The genome of viruses is DNA or RNA. We're talking about viruses, and those can be either DNA or RNA. Remember, they can't be both. So they can be DNA or RNA for viruses, for the genome. So chromosomes. Chromosomes are discrete cellular structures composed of DNA that's very packaged tightly together. Okay. So eukaryotic chromosomes are located in the nucleus and are multiple and linear. They're a long line of DNA. Bacteria chromosomes are usually a single circular loop. Remember, eukaryotic chromosomes are DNA double helix bound together. It's like a ladder, and then twisting that ladder around. And hydrogen bonds holding that together. In bacteria, it's usually just a single loop of DNA. Okay. You can see eukaryotic cells, you can see the chromosomes inside the nucleus. There's your chromosome, there's your nucleus. Remember, the nucleus is just the darker spot there. That's not your chromosome, it's not your DNA. You have mitochondria. Inside the mitochondria, they have DNA also. They have their own individual DNA, which supports the theory that mitochondria were once parasitic organisms that invaded the host and then just got incorporated into it as the organisms and the organisms evolved. Okay. And then bacterial chromosomes, bacterial DNA is circular. Okay. And viruses is just either DNA or RNA. That's usually single-stranded or double-stranded inside the cell. Okay. So chromosome is divided into genes. Genes are sections of DNA that code for a specific protein. Gene is the fundamental unit of heredity responsible for a given trait because it codes for that protein. You have a site on the chromosome that provides information for a certain cell function. You have a segment of that DNA contains a necessary code to make that protein from that RNA molecule. Your DNA converts to RNA, the RNA comes out in the cytoplasm and makes your protein. So there's three basic categories of genes. Genes that code for proteins, which make up all your enzymes, all the structures of your body, or those called structural genes. Genes that code for RNA, and genes that control gene expression, turn on or off these structural genes. They regulate whether these genes are active or whether these genes are docile. Those are three basic categories. All types of genes constitute your genotype. Remember we said genotype is a total sum of all the DNA. When you have these traits that are observable traits, This is called a phenotype. Okay, that's the expression of the genotype that creates observable traits from the proteins that are made. That is a phenotype. Okay. The size of it, the smallest virus is 4 to 5 genes long. That's the smallest you got. E. coli is a single chromosome that contains about 4,288 genes, roughly about 1 millimeter long. Okay, it's about 1,000 times longer than the cell itself. Because remember, it coils around. The human cell, humans have 46 chromosomes, 23 pairs, 23 from the mother, 23 from the father. Okay, they contain roughly 31,000 genes in the human genome. If you laid all the genetic material out, it'd be about 6 feet long. It's about 180,000 times longer than the cell. That's a lot of DNA in those cells. So the DNA molecule is compacted into cells by what's called supercoiling or superhelices. Okay, it coils around itself and condenses. So when you go to divide the cell, it's easy to split it apart. It can't be expressed when it's condensed. It has to be laid out open so it can be expressed and develop the genes that develop the proteins that it codes for. So in prokaryotes, by the action of the enzyme DNA gyrase, coils of chromosomes in a very tight bundle, reversible series of twists of DNA into the DNA molecule. Okay. So it twists them very tight, undergoes mutation, then it opens back up, that gene can be expressed or used. In eukaryotes, it's a little more complex. There's three-dimensional or levels. You have coiling, starting with chains of the nucleosome, and that's what DNA wraps around these proteins called histones. There's these little balls that the DNA wraps around and it allows the DNA to condense very tightly, and those form invisible chromosomes. Okay. So we have DNA, this with the double helix. Okay. Chemical tags attached to these histones. All these little balls here, these little balls are histones, and the DNA wraps around the histones. Okay. And it condenses and condenses further, and it condenses and then wraps around and condenses into a chromosome. Those chromosomes can have a center that can be pulled apart. Half of that's going to go to one cell, half goes to the other cell during mitosis. Then we got prophase, metaphase, anaphase, telophase, where they line up, metaphase and anaphase are pulled apart, and then the cell splits. Okay. So that's how DNA coils around itself, it uses histones in eukaryotic cells. Okay, the basic structure of DNA is a nucleotide. Remember, nucleotide is a sugar, phosphate group. It's a sugar phosphate background. That's what DNA is. So the sugar phosphate group is your basic unit. I need sugar. There is like adenine, guanine, cytosine, thymine, one of the nucleotide bases. And this bonds to another one. And this is what causes your double helix to form. Okay. So you have in DNA, it's a deoxyribose sugar. In RNA, it's a ribose sugar. So you have a ribose sugar, you have a phosphate group, and then you have nitrogenous base, which is either adenine, guanine, thymine, or cytosine in DNA. In RNA, you don't have thymine, you have uracil. It's one of the differences between DNA and RNA. There's 3 main differences you need to know between RNA and DNA. Okay, we'll go that in a little bit, we'll give you a little preview of it. RNA is single-stranded, DNA is double-stranded. RNA has a sugar ribose. DNA has a sugar deoxyribose. It's missing an oxygen. And instead of thymine in RNA, it's replaced with uracil. Those are the 3 big differences between DNA and RNA. Okay? But they're both built on this sugar phosphate backbone. And then nitrogenous base attached. And these are called nucleotides. Each of these sugar phosphate units with a base attached to it. Okay, so here's your sugar. And again, you have your phosphate, you have your whole backbone is going like that. And attached to the sugar is your nitrogenous base, which could be adenine, cytosine, or guanine. Okay, and it pairs across with adenine always pairs with thymine, cytosine always pairs with guanine. Okay, you always get A and T pairing together and C and G pairing together. Okay, A and T have 2 hydrogen bonds that connect them. Cytosine and guanine has 3 hydrogen bonds that connect them. So the middle of the DNA is connected by hydrogen bonding. The others are covalent bonding. So hydrogen bonding is not as strong as covalent bonding. So when DNA opens up, this is like a zipper, unzips. And then it exposes these bases so we can either duplicate it or express it for control of the gene messenger RNA. Okay. So nitrogenous bases covalently bond to the number 1 carbon of each base on the sugar and span to the center of the molecule to pair with a complementary strand. Adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. So when we start making bases by transcription from DNA, the first pair to open up is adenine thymine because that's easier to open. That's where we start. Then the cytosine guanine, it's only 2 hydrogen bonds, so it's easier for that to open up. And that's what a double helix is. If you're confused what a double helix looks like, think of a ladder and then take that ladder and twist it. That's a double helix. Okay. And as you can see, adenine always pairs with thymine and guanine always pairs with cytosine. Okay. And this is DNA because it's 1 double-stranded and 2 has thymine inside of uracil. Okay. You have what's called anti-parallel arrangements in 1 strand. So the helix runs from the 5 carbon to the 3. So 5' to 3' direction. And the other side is the exact opposite. Runs from what's called 3' to 5'. So it just depends where it's attached to the sugar, and they run what's called anti-parallel. They're parallel, but they're the exact opposite orientation, opposite runs. First one goes 5 to 3, then it's going to go 3 to 5. Okay? Every strand provides a template for exact copying of a new strand. So when it opens up those hydrogen bonds, we can duplicate it, we can copy it. Okay? The order of these bases, whether they're like it's adenine, cytosine, guanine, thymine, adenine, whatever that order is, that's your DNA code. That order is the code for what proteins are going to be produced by the cell. Okay. Again, this is our 3' to 5', and then the other is going to run 3' to 5'. This can run anti-parallel to each other. Okay, but you see each of the sugar phosphate backbone. This is their backbone right here, and the bases connect. Adenine to thymine, cytosine to guanine, thymine to adenine, guanine to cytosine. Okay. You always get adenine pairing to thymine and guanine pairing to cytosine. You'll never see cytosine pairing with adenine and thymine, or guanine pairing with adenine and thymine. That's how they always pair together. And they're held together by hydrogen bonding. Okay. So when you have to maintain this code during reproduction, otherwise when the cell splits to form a daughter cell, you have to copy the DNA exactly. So each cell has the correct DNA sequences to make the proteins they need. Okay. So constancy with base pairing guaranteed the code will be retained. It only pairs in a certain way. When the strands are opened up, each strand serves as a template to replicate into another copy of itself. Okay. Order the bases responsible for RNA protein synthesis for each phenotype of the organism. So the DNA order determines the RNA that is made. The RNA goes in the cytoplasm and determines your proteins that are made. These proteins that are made determine the phenotype. The phenotype is the expression of the genetic information of the organism. Okay. So your first question. In DNA, adenine is paired with what? What is adenine paired with?
With thymine.
Adenine pairs with thymine. And cytosine pairs with?
Guanine.
Guanine. Very good. Perfect. So look for thymine and guanine right there. So it should be C. And it is C. Very good. Perfect. All right. So the replication process. Replication occurs on both strands simultaneously. It would take a long time. They had to wait for every strand to go individually, each one just one at a time. We're starting in different areas, and then we're going to work together on each strand, and both strands are going to go simultaneously. Okay. So it's a semi-conservative process, which means one strand is open, and both strands are open, and then bonding occurs on both of those strands. So the parent DNA molecule is uncoiled. The 2 strands are separated, which expose the nucleotides to serve as templates. And those nucleotides are now open because those hydrogen bonds are broken. And then 2 new complementary strands are formed. And then each of those strands form together and create a duplicate of your DNA. So it opens up just like this, it unzips. And then the DNA that exists serves as a template for a new strand to form. Okay. All chromosomes are going to have a specific origin of replication. Okay. It's where replication is going to be initiated. Okay. The replication is AT rich. We say it's AT rich because adenine thymine forms a double bond, guanine cytosine forms a triple bond. So if there's a lot of adenine thymine bonds in there, it's going to be easier to open because there's only 2 hydrogen bonds versus 3. So less energy is required to separate the 2 strands, and the body likes less energy. So there's 2 replication forks. Excuse me. Where new DNA is being synthesized. Each contains its own set of replication enzymes. So you have enzymes that bond to the DNA that allow it to replicate, and both sides are being replicated simultaneously. So here you see your DNA unzipping right here. And then you got your starting points, and it's going to synthesize another band of each one. And each one's going to run anti-parallel, 3 to 5 or 5 to 3. Okay. So if we look at here, here's where we're starting. This is what it looks like. Here's your replication fork. Okay. Chromosome to replicate is unwound by what's called a helicase, which is an enzyme that opens up the DNA. Forms a replication fork, which means it opens up. Okay. And then two template strands are formed. Okay. The next step is the template for the DNA, which is blue. That's your DNA that exists. The red one is the one being made. Okay, the template is orientated 3 to 5. So then DNA polymerase, which the enzyme allows DNA to duplicate itself, it adds nucleotides in the 5-3 direction, which is the anti-parallel direction. Okay. And then synthesis. Okay, so it can be synthesized as a continuous strand. So it makes this long strand of DNA here in the anti-parallel direction. Okay. Then the template for the lagging strand runs in the 5-3 on the opposite side. Okay. And the synthesis occurs backwards on this one here as it comes around. Okay. Then we use what's called a primase, which is an enzyme that adds an RNA primer to direct the DNA polymerase. Okay, to create a segment, DNA polymerase 1 removes the RNA primers that have started opening it up, so complementary DNA can fill in the open sites. And then we join it by cutting with what's called ligase. Ligase the 2 strands back together. And that's how you replicate DNA. Oops. Yeah. So what we're going to we'll go through each of the steps here. So the chromosome to be replicated is unwound by a helicase, and it forms your replication fork. Okay, the template strand, which was our blue strand in this case, was 3 to 5. So our duplicate strand is going to use DNA polymerase 3 to add nucleotides in a 5-3 direction. So it goes towards a replication fork. Okay. And that's the new strand being synthesized. And the template for the lagging strand runs 5 to 3, which is opposite to the leading strand. So it makes a new strand of DNA. And it proceeds away from the replication fork. So before the synthesis of the lagging strand can start, a primase adds an RNA primer to direct the DNA polymerase 3. Once it starts, that RNA primer is removed, and then the DNA can replicate. This small section of DNA that's being replicated this are what's called Okazaki fragments. Okay. DNA polymerase 1 removes the RNA primers, fills in the correct DNA complements. Now that open site network is removed, and then the unjoined ends of the nucleotides must be connected by a ligase to connect and bring them together to form a continuous strand of DNA. Okay. So in summary, what we did was to make an exact copy of the DNA. It involves about 30 different enzymes. So it's not just a simple process. Okay. The helicase unwinds it, unzips the DNA basically, double helix. Then you have an RNA primer is synthesized at the over the origin replication by the primase. Then DNA polymerase 3 adds the nucleotides in the opposite direction. You have a leading strand and a lagging strand. The lagging strand is synthesized continuously in the 5-3 direction. The lagging strands synthesized in short segments, and then we link those together. And then DNA polymerase 1 removes the RNA primers and replaces it with the DNA to make the DNA code continue. Okay. So there's your replication for it. The DNA starts to unzip. Okay. When the replication for it meets ligases, it links the DNA fragments along the lagging strand. As the replication proceeds, only the newly synthesized strands move down, but the DNA stays up. Okay. When the forks have gone the full circle, a termination site shuts the replication down because it's duplicated down. Two circular daughter molecules remain connected briefly, but are nicked and separated by a helicase. That's in bacteria where they only have circular DNA. Okay, so you have your two forks when it starts to pull down. And this new strand pulls down, and then it nicks with a ligase here and separates those into two strands of DNA. Yeah, so you have a circular DNA, not chromosomes. Okay, so why must the lagging strand of DNA be replicated in short pieces? Okay. We don't have limited space, so you can use this up.
Yes. Is it C?
Yeah, DNA polymerase works only one direction. Okay, can only go one way. Perfect. Exactly right. Your DNA polymerase only links in one direction at a time. Okay, so it's in short pieces, and then we can just link them together. Perfect. Very good. So genetic information DNA is conveyed to RNA through what's called transcription. Transcription is a process of DNA goes to RNA. That's transcription. RNA goes to proteins. That's translation. Okay. So when DNA is converted into RNA, that's transcription. RNA, the proteins they form, is translation. Okay. So here's our DNA. This is circular DNA like bacterial DNA. Okay, we get replication of it. We get transcription of it goes into messenger RNA. You also make transfer RNA and ribosomal RNA. Okay. But the messenger RNA gets transmitted to the ribosomes, and the ribosomes make the protein using the transfer RNA to attach amino acids into a long chain. That's kind of the overview of it. So every structural gene is an ordered sequence of nucleotides that codes for a protein's primary structure. Remember protein's primary structure. Remember there's primary, secondary, tertiary, and quaternary. Primary structure is just the line of amino acids linked together. Secondary is when they start to form an alpha helix or beta pleated sheets, start to form together. The tertiary structure is when they form the three-dimensional shape, which makes them active. And the quaternary form is when more than one of these tertiary proteins come together to form a functional unit. So that's what the genes determine that sequence. There's a group of 3 consecutive bases called triplets, okay, or codons. Those code for a particular amino acid. And we'll show you the codes for each of those. So on one strand of DNA, they're transcribed into RNA sequence triplets. Each triplet of this nucleotide in RNA specifies a particular amino acid. So every transfer RNA has a particular amino acid bonded to it. So the transfer RNA is going to bond to the ribosome that is reading the messenger RNA to bind to the triplet code. We'll show that works as a little diagram come up here. So the protein's primary structure determines its shape and function in the future, because it's going to fold according to hydrogen bonding, hydrophobic, hydrophilic areas, sulfur, disulfide bridges, things like that, are going to contribute to or control its three-dimensional shape. So these proteins contribute to the cell phenotype. Maybe your phenotype is the expression of the cell, okay, characteristics of the cell. Okay? They contribute to the cell's phenotype as enzymes and structural proteins.
Professor, isn't a phenotype, can it be like eye color, hair color?
Yep. Shape of your ears, no, yeah, all that. That's an expression of your genetic material. Anytime you have an expression of material, that is a phenotype. Exactly right. So here's DNA. Okay, there's our DNA code. Remember, DNA, guanine pairs with cytosine, adenine pairs with thymine, etc. This opens up, this unzips, and then this is the complementary code of RNA to that top line of DNA. CUG, remember here's guanine pairs with cytosine pairs with guanine, uracil now because it's RNA. It's not DNA, so you don't get thymine anymore, you get uracil. And C pairs with G, T pairs with A. So all these are coding. So this G pairs with this C, this A pairs with uracil, C pairs with G, T pairs with A pairs with uracil, T pairs with A, and so on all the way down. Does that unzip and open up now your messenger RNA? Each of these three, let me get rid of these codes here. Each, I didn't work very well, did it? There we go. Each of these three here is what's called a triplet. Okay. This code is read as a triplet form. The transfer RNA has three matching triplets. So your transfer RNA kind of looks like, it's kind of funky here, but it would have G, A, C, and the GAC would bond right to this, to the cytosine, the uracil, and guanine. And GAC on a transfer RNA card codes for leucine, your amino acid. So when the ribosome reads this, it matches it with this code here. These triplets combine, and we put leucine and links that amino acid to the chain. Okay, we have AUA. The next one, it's going to code for uracil, adenine, uracil. That's the transfer RNA. You're going to code here. It's going to match up with that. And then the ribosome is going to stick together another amino acid. This is going to be isoleucine. So 3 unit codes code for our particular amino acids. And there's only 20 amino acids that make up all the proteins in the entire human body. Okay. General structure of ribonucleic acid RNA is different than DNA. We talked about it a little bit, remember? RNA is single-stranded, DNA is double-stranded. We can have messenger RNA, transfer RNA, and ribosomal RNA. RNA contains uracil instead of thymine. DNA contains thymine. And the sugar in RNA is ribose, okay? Rather than deoxyribose. It's missing an OH group. It's missing that oxygen. That's why it's a deoxyribose. So RNA types. Remember we said we have a messenger, transfer, and ribosomal. Messenger controls our sequence of amino acids and proteins. It's determined directly from the DNA. And translation. So this is the only one that's translated is messenger RNA. Transfer RNA gives you specific amino acids. It's a triplet code, and it carries amino acids to the ribosome where it's stapled together to make a long chain of amino acids. Ribosomal RNA is found, okay? Those are large structural ribosomal RNA molecules. Okay? They form the major part of ribosomes and participate in protein synthesis. Okay? And then a primer that can help you begin DNA synthesis, and then is kicked off and then spliced back in with DNA nucleotides. So it kind of gets the DNA started. It primes it for replication. So messenger RNA. Messenger RNA is a transcribed version of the gene in DNA. So if your DNA, if we unzip our DNA and we end up with adenine, I don't know why I put an M there. Adenine, thymine, cytosine, guanine, cytosine, adenine. That's unzipped now. So the messenger RNA is going to come along and it's going to bind here, and it binds the code of uracil, adenine, guanine, cytosine, guanine, uracil. So this is now the code of messenger RNA. This code goes out of the nucleus and to the ribosome, and then is coded into triplets where transfer RNA binds to each of these triplets here and attaches amino acids according to this code that's been translated. Okay? And this is called codons. Those are triplet codes. Okay? So here's your messenger RNA. And each of these triplet codes is going to bind to a particular transfer RNA, which is going to give you a particular amino acid to add to the chain. Okay. So transfer RNA. Transfer RNA is what controls your amino acids. It binds to them. Okay, it's a translated messenger RNA code into the proteins because it adds the amino acids specifically together. It's about 75 to 95 nucleotides in length, and it's bent into a hairpin loop. Kind of looks like a clover like that. And then this binds your amino acid right here. This is your amino acid binding site. Okay. So the bottom loop of the clover leaf exposes the transfer RNA, the anticodes to your messenger RNA. So if your amino acid's here, your 3 codons are right here. Okay. So now that was your transfer RNA. Okay, the transfer loop loops back to itself to form this interaction chain, hydrogen bonding. That's what forms its shape. It's that cloverleaf structure. Okay. The bottom is the anticodon that attaches to your messenger RNA and then allows your particular amino acid to attach. Okay, and that's what they look like. This is your hairpin loops here. This is the anticodon. This is the 3 triplets that bind to the messenger RNA. So here's your messenger RNA right down here. And then here is your attachment site for your amino acid. And it depends on what these 3 codons are, determine what this amino acid here that's attaching to the chain. Okay. So this is your ribosome. Ribosome is basically the stapler that staples these transfer RNAs together and allows this protein chain to exist. So this is your messenger RNA right here. Okay. This strand here is your messenger RNA. These are the transfer RNAs that bind to it. As they bind to it, they get passed along, and then this is your amino acid that's attached to it. And this gets transferred to this, and then this gets kicked off. And it starts to make an amino acid chain as each of these transfer RNAs bind. Remember, the ribosomes have a large subunit and a small subunit. And in between it, that's where that messenger RNA chain runs. Okay. So during transcription, RNA molecule is synthesized. Perfect. Okay. There's 3 stages. You have initiation, where the RNA polymerase binds to the promoter region. Okay, upstream of the gene. Then you got elongation, where the RNA polymerase adds nucleotides that are complementary to the DNA strand. Remember, if it's adenine, it's going to form with uracil. Cytosine is going to form with guanine, and vice versa. Okay, and then you got termination, has to stop it. Once it makes a messenger, it has to have a stop code that stops at a particular sign. Okay, and depending how long it is, 100 to 1200 bases long, depending what protein is being coded. Okay. All right, so each gene contains a specific promoter region, okay, and a leader sequence to get it started. This guides the beginning of your transcription, how you take your DNA to your RNA. Then you have your gene that codes for the polypeptide chain, which has terminal sequences that stop your translation. Okay, the DNA is unwound. Remember, it unzips itself at the promoter by RNA polymerase. That's what gets it started. It opens it up, gets your DNA fork opened up. So only one strand of DNA, called a template strand, supplies the codes that are transcribed by RNA. So RNA is only going to bind to one side that opened up. It's not both sides of this one. And the strand it binds to runs in a 3 to 5 direction. Now this RNA polymerase moves along the DNA strand that adds your complementary nucleotides, okay, as dictated by the DNA. Messenger RNA strands reads in a 5 to 3 direction. So the polymerase continues transcribing until it reaches a termination site. The messenger RNA transcription released, okay? And that section of DNA is now rewound up back and the fork is gone to its original configuration. So it opens up, allows the RNA to do its thing, kicks it off back in the cytoplasm to do its function, and then closes back up when it's done being used. Okay, here's a summary of what we just went through. It opens it up. This is your DNA. Then it opens it up, and then the RNA reads that top strand. You get complementary strands. Okay. Opens it up, and then as it reads it, this section closes back up when it's done. Okay. And this creates a messenger RNA strand, which is complementary to the DNA strand that it had. This messenger RNA feeds into ribosomes. These ribosomes link the transfer RNAs, these transfer RNAs here, where you have your amino acids, and your codon is here, and it's bind into here, and it passes amino acid to the next one and builds a chain of amino acids, which is your primary sequence for your protein, primary structure. Okay, so you can see, here's your amino acids. Okay, here's your messenger RNA, and this is a codon, the triplet code that is binding to the messenger RNA to determine the order of your amino acids. So the messenger RNA that comes from the DNA determines the order of amino acids, which determines the shape of your proteins, which determines what protein is being made and the function of that protein. So all the elements need to be synthesized protein, messenger RNA, transfer RNA, amino acids are brought together on the ribosomes. You can see that right here, large and small subunits. It occurs in 5 stages. You get initiation, elongation, termination, and then you get the protein folding and processing, going from your primary protein structure to your secondary, to your tertiary, to your quaternary. The genetic code, your DNA code, represented by the messenger RNA codons and their specific amino acids. So each codon represents specific amino acid. Remember, there's 20 amino acids in the human, and 20 amino acids, they make up all the proteins. So this code is universal among all organisms and is redundant, which means more than one sequence can code for the same amino acid. But all organisms use the same code. They're all the same. And this is the code right here. If the codon is UUU, it's going to be phenylalanine. UUC is also going to be phenylalanine. Serine is any of these 4 codes. So if you see the code, you can tell what amino acid sequence is going to occur. AUG is a start sequence. Okay. UAA is a stop sequence. And you can see there's redundancies built in. So in case there's an error somewhere, it can be fixed with a redundant code. Okay. That's basically how it works. So transcription produces messenger RNA that's complementary to the DNA strand. So during translation, transfer RNA use their anticodon to interpret the messenger RNA codons and bring in the specific amino acid. So this is your DNA. It opens up. RNA is your complementary code to this DNA strand. This complementary code binds your, in sets of 3, binds your codon. Okay. And then this codon carries a specific amino acid. Okay. As it goes through the ribosome, as they bind together, it transfers these amino acids into a chain and kicks it off the transfer RNA. So you get a chain of amino acids linked together. Okay. So the ribosomes assemble on 5' of the messenger RNA transcript. Ribosomes scan the messenger RNA until it reaches the start codon, okay, which is usually AUG from that chart that we saw. Then a transform RNA molecule with its complementary anticodon, okay, enters the P side of the ribosome and binds it to the messenger RNA, and that's what gets the process started. Okay, remember, UAG was a stop codon. There's your start codon, it binds to it. The messenger RNA and the transfer are brought together, and then it's going to read it going across the direction here. And then this amino acid is going to be transferred to this and then kick it off to form a chain. So that's how they start with the transfer RNA. Okay. And it starts reading each codon, its complementary pairs. Okay. Then these amino acids bind to each other with a peptide bond. Okay. And then this transfer RNA is kicked off, leaving its amino acid behind. And it's going to do the same process like a stapler, over and over. And it's going to create a chain of amino acids. Here's the third one coming in. It's going to bond to this site, and it's going to add this amino acid to the protein chain. Okay, there's your peptide bond being formed, your second one. Okay, and then here's your chain being formed. Fourth one comes in, reads this codon, reads the next codon, codon, etc., and it transfers the correct amino acid to your chain, depending upon the codon, the codec being read in the triplicate form. Right, the fourth one binds to it, and you get back, and then you're going to get, whether it's 100 times, 1,000 times, you're going to get this long chain of amino acid. That determines this is your primary protein structure and chain of amino acids. Okay, until it reaches a termination codon. You got a start codon, which started the ribosome to bond to it. Now you got a termination codon, which is one of these three, UAA, UAG, or UGA. There's no corresponding transfer RNA, it doesn't bond it. So it hits that codon, no transfer RNA can bind to it, it stops. Okay. When they reach that codon, the ribosome falls off, breaks apart into two subunits. Okay. And the last transfer RNA is removed from the polypeptide, creating a primary structure polypeptide chain. All right. Next check. Transfer RNA is a molecule that does what? Contributes to the structure of ribosomes, acts as a transcription of DNA, transfers the DNA code to messenger RNA, or acts as a translator messenger RNA code into proteins. What does tRNA do, transfer RNA? Which one best describes it?
Is it B?
I mean, transcription is a messenger RNA going to DNA go to messenger RNA. So transfer RNA binds the amino acids together on the ribosome and forms your protein.
Oh, so it'd be D.
It'd be D, because if that's what's actually forming your protein, it's this chain, this link of amino acids as these tRNAs, the transfer RNAs bind to the messenger RNA code in the ribosome and kick its amino acid into the chain. So it should be D, yeah. Acts as a translator messenger RNA code into proteins. That's what forms your protein, is your amino acids. Does that make sense now?
Yeah, it does, because B doesn't make sense because that's not the transcription of DNA.
Right, that's what messenger RNA does.
Yeah.
The transfer RNA does your actual forms your protein by linking your amino acids together. Back to polyribosomal complex. Again, this is your RNA. Okay. RNA is being read and it goes into transfer RNA, and this is your messenger RNA that's continued from the DNA here. Okay, these are your ribosomal units, and you can see how the transfer RNA are attaching and then transferring their amino acids, and it grows this long chain. That's how it works. And this is a scanning electron microscope picture of it. You can see how the amino acids are forming along the ribosomes, and it just forms their chains. Eukaryotic transcription and translation. This does not occur simultaneously. Transcription occurs in the nucleus. Okay. Translation occurs in the cytoplasm. Transcription is DNA going to messenger RNA. The messenger RNA moves through the pores in the nuclear envelope to go to the cytoplasm. In the cytoplasm, you have your ribosomes. Your messenger RNA is transferred to your amino acids by your tRNA, transfer RNA. Okay, so transcription occurs in the nucleus, translation occurs in the cytoplasm. So in eukaryotes, the start codon is AUG. Okay, a messenger RNA encodes a single protein. Okay, so every RNA does a single protein for eukaryotic cells, but bacteria, they can put multiple proteins because only that's circular DNA. Eukaryotic DNA contains what's called introns. They're intervening sequences of non-coding DNA. So there's sections inside your gene here that don't code for anything. These are called introns. They're in between. Okay, they have to be spliced out of the final messenger RNA transcript. So before the RNA is used, these are spliced out of the way so these can come together. Okay. So eukaryotic sequence of what's called exons and introns. Okay, the introns are moved out and the exons are spliced together. Introns are transcribed but not translated. Okay, they don't form, they're removed before the transfer RNA gets to put the amino acids in place. Okay. Splicing does not occur in prokaryotes. They don't have this. Only eukaryotes have the exons and the introns. So this is your exon, which is your section of coding, and then your intron is your non-coding section. Okay, so this is a DNA template. Then you get your primary messenger RNA, which codes over to the introns and the extrons. And then the introns are like a lariat and they're forced out. They're looped and they're cut out. Okay, this puts the exons together, having a functional messenger RNA. So the messenger RNA from the nucleus is modified before it binds to the ribosomes by removing the introns, and it links the exons together, or the coded units. So how do we regulate these genes? I mean, it doesn't make sense for the body to produce, say, insulin when there's no sugar in the bloodstream. So we regulate, we produce the proteins that we need for the body. So genes are regulated to be active only when their products are required. In prokaryotes, it's controlled by what's called operons, which is a set of genes, and it's regulated as a single unit. It's that one circular loop. Okay? There's two types of operons in prokaryotes. There's inducible and repressible. Inducible turns it on. Okay, when the enzyme needs to metabolize something, the nutrient, then it's produced when it's needed. Turns this gene on. Okay, they're turned off, repressible, when there's no substrate around for that enzyme to work on. Okay, so those are the two types for a prokaryote. Okay, there's three systems. There's a regulator, which is the gene that codes for repressor. There's a control locus, which is composed of promoter and an operator to trigger the signal of the gene to go on. And there's a structural locus, which is three genes that code for an enzyme to catabolize lactose, which is sugar. Okay. Beta-galactosidase hydrolyzes lactose. Permease brings lactose across the cell membrane. And beta-galactoside translase, they're not quite sure what it does, but these three enzymes are turned on when lactose is present in the bacteria. Okay. So the lactose operon is normally turned off. So in the absence of lactose, okay, repressor binds with the operator locus and blocks transcription of the DNA of what comes downstream to the structural gene. So it doesn't make the enzyme necessary to break down lactose because it's not present. Okay. So the operand is off when there's no lactose present. There's your operator right here, and it binds. So it stops transcription of that DNA. Okay. So in the absence of lactose, a repressor protein, okay, attaches to the operator of the operon. You can see it attaching right here, and it stops this from occurring. Okay. This blocks the transcription of structural genes going down from this point. It can't happen. It's sitting right in the way. Okay. So suppression of transcription and then forward and translation, because transcription is not occurring, prevents unnecessary synthesis of enzymes for processing lactose if lactose isn't present. Now, when lactose is present, it turns on the operant gene by acting as the inducer. Okay, the binding of lactose to the repressor protein changes its shape, and it causes it to fall off the operator. So RNA now polymerase can bind to it and turn the gene on in the presence of lactose. So upon entering the cell, this lactose becomes a genetic inducer. Okay, so it attaches to repressor, makes it inactive, and the repressor falls off. Now the DNA can be accessible to the RNA polymerase. RNA polymerase transcribes the structural gene, and the messenger RNA is translated into enzymes that can act on the lactose substrate. Okay. So lactose is present. Here comes lactose. It binds to our repressor here, and it changes its shape and makes it fall off. Now the RNase polymerase can become active, and we can start transcription of this gene of DNA. We make our messenger RNA, it goes to the ribosomes. It makes and goes to transfer RNA into the ribosomes and makes our protein. Okay. Arginine operator is a repressible gene. It's normally on. It's the opposite of the lactose one. And it turns off when the product is no longer required. Okay. So the operand is on for arginine, and it's being used by the cell. Okay, the repressible operand remains on when its nutrients products, this is arginine in this case, are in demand by the cell. The repressor has the wrong shape to bind the DNA without its co-repressor. The co-repressor is the prensyl. Once it makes enough arginine, then the arginine acts as a co-repressor and stops it from, and shuts the gene off basically. So RNase is free to and actively transcribe the gene, and translation actively proceeds until the levels of arginine build up. Then that is the co-repressor that binds to the repressible operand and stops the process. Okay, so this is normally what happens, and you're making alanine, arginine, I'm sorry, arginine, ARG. Okay, so we normally make this as the cell goes on. When the arginine levels build up, okay, it's a co-repressor. It bonds to the normal repressor, changes its shape, and then allows it to bind to the gene, and then it stops it. It stops the production. So it's normally turned on. When this builds up, it goes back, binds repressor, and this binds here and shuts that down. Okay, so when excessive arginine is present, okay, it binds to the repressor and changes its shape. And that blocks the arginine synthesis. So the arginine itself, the product, is the co-repressor. So arginine build up, operand is repressed. When arginine builds up, it serves as a co-repressor that activates the repressor. It binds to it, changes its shape, allows it to bind to the DNA strand and stop the RNA from transcription. Okay. So your activated complex affixes to the operator and blocks the RNA polymerase. So further transcription of the gene for RNA synthesis is blocked. It cannot occur. So see how when the arginine builds up, it binds to the repressor, changes its shape, and allows it to bind to the DNA. So arginine synthesis has to stop because the RNA polymerase can't work anymore because this blocks it. Okay. So if an operand's repressor is in its active form, that means what? Transcription for the operand is occurring, or transcription for the operand is not occurring. If the repressor is in its active form,
It's occurring.
the repressor, operand's repressor means it stops it.
Sorry.
The active form of stopping it means it's not going to occur. Does that make sense? This is a repressor, and when you activate it into its active form by the arginine, whatever is being produced, then it's going to repress the gene. So the operand is not, transcription is not occurring, so you don't get translation and get the protein being made. Does that make sense?
Yeah.
Sort of, okay.
Yeah, it does. I misread the question.
Okay, oh, no problem. All right, mutations. Mutations in genetic code, things that change. So a change in phenotype, due to change in genotype, is called a mutation. When one base is either switched or skipped or something like that, we have a mutation when the DNA is an exact copy. Natural non-mutated characteristic, what's called the wild type, which is what you normally occur. But when an organ has a mutation, you get what's called a mutant strain, showing various either morphology, nutritional characteristics, genetic control mechanisms, resistance to chemicals, etc. It's just like bacteria. When we feed them antibiotics, okay, they become resistant because bacteria replicate at a much faster rate than we do. So their replication rate, their mutation rate is much higher. So when they get a mutation, one of those mutations can be resistant to penicillin. So we can select for bacteria that are resistant to penicillin. That's where there's this big push not to give drugs to everybody, because we're getting these super infections we can't treat anymore. Right? Because we give people antibiotics. What's that?
Like superbugs, because the body becomes resistant to it.
Well, the bacteria become resistant to the antibiotics we're using. So if you get a bacterial infection and we treat it, the bacteria are going to become resistant because bacteria evolve, and they evolve at an extremely fast rate. And they have mutations, and some of these mutations are going to be resistant to the antibiotic that we use. So then if we keep using antibiotics, and we put antibiotics in the feed we give for cows, things like that, we're selecting bacteria that are resistant into the population. So we're selecting bacteria we cannot treat with antibiotics. And that's the problem we're running into. Okay. So these mutants, a mutation can be bad, and then, or it could be good. Could give you an advantage. Okay. So mutations are a way of changing our DNA one way or another. So we have, when isolating these mutants, the replica plating technique allows us to identification of different mutants or exposure. So culture is exposed to a mutagen, something that causes a mutation. Radiation, sunlight, get all kinds that can cause electrical interferences, and they can cause mutations. Isolated colonies are transferred to a master plate. Tiny clumps of cells are then picked up and transferred to a plate with a complete medium and other incomplete medium. And the colonies in the complete medium, missing from the incomplete one, are mutant colonies that can be subcultured for further use. We can identify which colonies have a different genetic makeup by transferring to a different medium. Okay. A spontaneous mutation is random occurring change in DNA due to errors in replication that occurs without any known cause. There's sometimes the wrong transfer RNA is going to bind to it just by freak accident, and you're going to get the wrong amino acid. There's just a spontaneous mutation that occurs just randomly, no known cause. Induced mutations, those are exposure to known mutagens. Okay, primary radiation, chemical agents, okay, that disrupt the DNA. Okay. The different chemicals can cause different types of mutations. Nitrous acid can remove an amino group, it's nitroxidation. Ethidemium bromide inserts between the paired bases. Okay. Nitrogen-based analogs can cause a complete with natural bases for cellular obtained DNA. They compete with those sites. Radiation, ionized radiation like gamma rays using X-rays. Okay. Cause single or double breaks in DNA. Ultraviolet radiation, tanning booths, cause cross-links between adjacent pyrimides. Okay. There's different categories of mutation. A point mutation occurs at a specific point on the DNA. It could be an addition of one extra amino acid, could be deletion of one nucleotide, or it could substitute one for another. Missense mutations cause a change in a single amino acid. Nonsense mutations cause a normal codon into a stop codon. Okay. So instead of contributing protein, it stops it. This is usually a fatal error. This usually destroys a protein because it's not where it's made. Silent mutation alters the base, but does not change the amino acid. Remember, we have redundancy in our code. So sometimes AUG and AUC code for the same thing. So if we get a mutation and it turns from a G to a C, but it codes for the same code amino acid. So this sign doesn't make any difference. A back mutation is when a mutated gene reverses to its original composition. You get a mutation, then it goes back. That's a back mutation. Or frame shift mutation. So when reading the frame environment is altered. So it shifts how it's read. And then that codon, instead of reading CAG, AUC, instead of reading that as your codon, now it could read this as your codon, AGA. It shifts it. Instead of reading this here, it reads those three. That's called a frame shift mutation. Okay. Wild types are non-mutated. That's what we see in our environment. The big bad cat ate the fat red bug. Okay, there's your genetic sequence. Okay. That's your what's considered quote normal sequence of DNA. Nothing's mutated. That's what your wild sequence is. Substitution, it substitutes one a nucleotide. Okay. A missense mutation causes a different amino acid to be incorporated into a protein because it changes the triplet code. Okay. The range from it could be unnoticeable or it could be severe. Depends on how the amino acid alters the protein function. It's going to change the shape of it. Okay, in the nonsense, it puts a stop code on. So the rest of this protein never gets made. Okay, this is premature termination of the protein. It's almost always severe because it stops the protein from being made. It's almost always stops the function of the protein. Inversion. This inverts the letters. Okay, so it allows this to switch with this. It inverts the two ways here. You get inverse mutations. Okay. So inversion is derived when adjacent letters are exchanged, switch places. It alters one for the other. Depends on the location of your inversion. Does not change the reading frame, but can result in a significant change if these amino acids are going to be different. Remember, you got a redundancy code. Okay. Frame shift mutations. These are really bad. Okay. You start to read all that across, and then it shifts by deletion here. And all these ones are shifted one to the left. So the original codons are not the same. This could be an addition of a letter or deletion of one. Okay. It shifts your reading frame. Resulting protein, which every amino acid can be affected. And so after the mutation, every one of these amino acids can be affected. It almost always results in a non-functional protein, just like a stop codon. Usually gives you a fatal error. How can we repair mutations? Okay. Since mutations can be potentially fatal, the cell has several enzymatic repair mechanisms to find and repair damaged DNA. So DNA polymerase, one of our enzymes, proofreads nucleotides during DNA replication to help make sure we don't miss one and get one off that we're not supposed to. It checks it. We have a mismatch repair. It locates and repairs mismatched nitrogen bases that were not repaired by the DNA polymerase, like a backup system. We have what's called light repair, for where UV light damages our DNA. We have excision repair, which locates and repairs incorrect sequence by removing that sequence and adding the correct nucleotide sequence back in. So you have some ways that are limited that can repair certain kinds of mutations. We have what's called the Ames test. Okay, it's a chemical capable of mutating bacterial DNA, and it can mutate mammalian DNA. Okay? Using agriculture industry, medical compounds are screened using this Ames test. Okay, the indicator organism is mutant strain of Salmonella typhimurium that has lost the ability to synthesize histamine. Mutation is highly susceptible to what's called a back mutation. You refix that and put it back to the original form that it was in. Okay, but you take your culture, you use a control plate and a test plate, okay, incubate it, and you have certain histology colonies, spontaneous back mutation, and certain ones that don't, and then you can regroup, re-inoculate those colonies and see which ones are missing that, or which ones are exhibiting that mutation. Mutations leading to non-functional proteins are harmful and potentially fatal to the organism. Organisms with mutations that are beneficial in the environment can be readily adapted, survive and reproduce, just like bacteria that are resistant to penicillin. Okay, these mutations are basis for a change in populations. Any change that confers an advantage, okay, will be retained by the population. If it's disadvantage, it's going to disappear from the population. Okay. Which of the following mutations would cause a frame shift mutation? In other words, reading it, it would shift over. Can't have T. In other words, it's read that, it would shift. So it reads that one and that one. Okay. Which mutation would cause a frame shift? A silent mutation, a missense mutation, a nonsense, or deletion mutation? One of those would shift one letter over. Which one would be it?
Is it the, I want to say it's B
Now, missense would just be,
or D or D.
I'd say D, because if you delete this one, then you've shifted all that over. A deletion or an addition mutation. Either one of those are going to cause a frame shift because there's going to be an extra amino acid or there's going to be one less amino acid. So either way, it's going to shift that reading to have a different trip code on code. So a deletion mutation or an addition mutation would cause a frame shift. So in this one, it would be delete, it would be D. Genetic recombination occurs when an organism acquires and expresses genes that originate in another organism. So a gene in one organism is transferred to a DNA in another organism. There's three means for genetic recombination. There's what's called conjugation, transformation, and transduction. Conjugation. Conjugation is a transfer of plasmid. Remember, bacterial DNA is a circle plasmid. Okay. Transfer of plasmid, a chromosomal fragment from a donor to a recipient by direct contact. So two cells come into contact and you directly transfer DNA material from one to the other. Gram-negative cell donor has a fertility plasmid called F factor, allows the synthesis of a conjugate, what's called a pilus. The donor, which is an F positive cell, transfers the fertility plasmid through the pilus, which is the extension, to the recipient cell, which becomes an F plus cell now because it's received new DNA. Some F plus cells become HFR cells, high frequency of recombination. It's a lot of recombination is brought into the cell. So it's a physical connection, makes contact with the cell. These two cells are now in contact, F plus and F minus, and then it's transferred. Okay, this is transferred from one cell up into the other. And now you have the F plus cell. So the pilus of the donor cell on the top attaches to receptor of the servant cell and retracts to draw the two cells together. So this attaches, your pilus attaches, and it's like a spring, it pulls them together. Once it pulls them together, now this genetic material can be transferred from one to the other. Okay? That is conjugation. So high frequency recombination, when the donor's fertility plasmid is integrated into bacterial cell's chromosome. So when this actual integrates into the circular chromosome. So when conjugation occurs, a portion of the chromosome and a portion of the fertility plasmid are transferred to the recipient. So now you've got this transferring, which is one, but here's your attachment here. And then you can see the DNA moving in here. And you got a complete transfer of this circular plasmid of DNA from one to the other. So you have a transfer of that factor of the conjugative plasmid. Okay. Or high frequency transfer. Okay. You can see how it transfers around. This gene transfers across once the plasmid connects them, pulls them together. You see how this transferred, and then this actually incorporates into the existing genome of it, into the existing plasmid. Okay, it involves a transmission, that's a high frequency transfer. It involves a transmission of a chromosomal gene from a donor cell to a recipient cell. The donor chromosome is duplicated and transmitted into part of the recipient cell. It gets integrated into the chromosome itself, and therefore every time that cell replicates, now that new gene goes with it. It's high frequency because every time that daughter cell is going to have that gene with it. Okay. Transformation. Chromosome fragments from a lyse cell, okay, are accepted by every recipient cell. Genetic code of the DNA fragment is acquired by the recipient. Okay. So donor recipient cells can be unrelated to each other, and it's used in recombinant DNA technology. We break it up and it absorbs it from the environment outside itself. So here's your transformation. Here's your receptor from the outside. It brings it into the cell. So this other cell lyses or however, and this is released into the environment around the cell. Okay, then it's pulled in, it's incorporated into the DNA. Okay, the recipient is now transformed the gene for synthesizing the capsule. So this is now part of the DNA and is replicated into every future generation. Okay, so the DNA fragment, which was blue in the slide before, delivered what's called a cap gene, which is the capsule. Okay. Binds to the surface, and the recipient cell pulls it in. DNA is converted to one strand, transported into the cell by the DNA transport system. DNA strands incorporated into the recipient chromosome. Okay, so now you have a gene for synthesizing a capsule. And that's how we do recombinant DNA. We put our cells out, the cell dissolves it in, and we can place that directly into the DNA itself. This is different works on these types of transformations. You got different cell types. So we have a live S strain, give it to rat, rat dies. You have a non-capsule, which is a live R strain, the rat survives. When you combine them, okay, you heat kill the S strain, give it to the rat. If you combine the larva A strain, even with its heat kill, even though both these by themselves do not cause death, both together cause the rat to die. Okay? The chromosome, the genetic material is transferred from one through transformation into the heat killer S strain. And now you got a live strain of 3 generations, and that reproduces the bacteria. And that allows it to kill the strain through transformation. Transduction. Remember, bacteriophages are viruses, right? We talked about that. Bacteriophage is a virus. So there's a carrier of DNA from a donor cell to a recipient cell. Remember, a virus, little funky things come down, and viral particles here, viruses inside, and binds to the cell and whoosh, injects that into the cell. Okay. You have generalized transduction or specialized transduction. Generalized transduction is random fragments of disintegrated host DNA are picked up by the phage during assembly, and the genes can be transmitted through the phage as an injection of the cells. Or specialized transduction. Highly specific part of the host genome is regulated, incorporated into the virus. So a specific part of the host genome is put back into the virus, and that can be transmitted. Okay. So here's your phage DNA, injects it in. Okay, it becomes part of the makeup of the cell, and you make more viruses, cell lysis, and these viruses are released. Okay. So the DNA released by the viruses are now incorporated into the cell. Okay, the phage is incorporated into the cell here. It can make the viruses from the cell DNA. Viruses are released. And then you have a separate viral package there. Okay, there's 2 different outcomes that can occur with that one. That can be either incorporated into it, just like the generalized, or it can have a special one outside of it, depending how the virus duplicates itself. So these are different types of recombinant DNA. You have the conjugation we talked about, transformation, and transduction. Okay. Remember, conjugation was direct contact. The other 2, transformation and transduction, are indirect because it uses a lyse bacteria cell or a virus to transfer that DNA into the cell. Okay. So direct means that they're in contact during exchange. Indirect means they're not in direct contact. Either take it up from the environment or take it during the phage. So special DNA segments have the capability of moving from one location in the genome to another are called jumping genes. They can move from one location to another location. They rearrange genetic material. Can move from one chromosome site to another. So it forms a chromosome to a plasmid, or from a plasmid to a chromosome. So it can pass from the plasmid to a chromosome material, or one way to the other. Could be beneficial, produced in the environment, or it could be harmful to the organism. So this section of gene can move to a different section. It can duplicate to multiple parts, or it can duplicate itself to a plasmid and then leave the cell and transfer to something else. Okay. Animal viruses. Okay, the viral genome is one or more pieces of DNA or RNA. Remember, one DNA or RNA contains only genes needed for production of new viruses. Remember, it doesn't have any genes to make anything else, any proteins, because it is only going to make new viruses. It's going to take over the cell's mechanism to use it for itself. So viruses require access to a host cell's genetics and metabolic machinery to instruct the host cell to synthesize new viral particles. Okay, most viral particles contain normal double-stranded DNA or single-stranded RNA, but it can contain single-stranded DNA or double-stranded RNA also. It's a little bit rare. Okay, but it's either one or the other. You can't have both, one DNA or one RNA. With few exceptions, replication of viral DNA occurs in the nucleus. Remember, DNA is found in the nucleus, and RNA viruses occur in the cytoplasm, where messenger RNA goes to make proteins. In all viruses, viral messenger RNA is translated into viral proteins on host cell ribosomes. Remember, the virus does not have its own ribosomes. It uses a host cell support, and it uses the host transfer RNA mechanism. That's what we talked about. In the same way, but it makes viral proteins, not cell proteins. So it takes over the cell mechanism with its virus. Okay. So viral DNA, it's injected here, produces viral messenger RNA, and this comes into this is in the cell. This comes into the nucleus. Okay. And this is messenger RNA from the virus, uses the cell's ribosomes to make viral proteins now, using it the cells. They make his own messenger RNA using the cell's transfer RNA. Okay. The viral DNA can get incorporated into the chromosomes. Okay. And it gets replicated along with the DNA. Okay. And it can come back in and unzips it, and it can make mature viruses when the viral proteins be assembled. Okay. So there's it can lay inside the DNA, less like the herpes virus. It can lay inside your nerve tract, incorporating your nerve cells, only come out when it expresses itself. Single-stranded RNA viruses, that was your double-stranded DNA virus, goes into the nucleus and does that. Now the single-stranded RNA viruses, they do not enter the nucleus. They stay in the cytoplasm. Your viral RNA goes in to converts it into messenger RNA. This reads it or uses itself, its RNA now, is read by the ribosomes from the cell to make viral proteins. Okay, these viral proteins can duplicate themselves and form the different capsids and stuff needed to make the new virus particles. Okay, so single-stranded RNA occurs in the cytoplasm. Double-stranded DNA occurs in the nucleus. Which of the following terms describe the genetic transfer of bacterial DNA that occurs by direct cell-to-cell contact and is mediated by bacterial plasma? Which one was direct contact? Transformation, transduction, or conjugation? The three different ways.
A.
Which one?
A.
Transformation is brought in. Conjugation is when they both conjugate. They put them together and they transfer allow it. That's conjugation. So it should be B. Yep. Okay. The other ones are transductions when your virus particle spread, or transformation when it's transmitted by one cell lysing and then the gene enters into the cell, pulled into the cell. But conjugations when they actually fuse together and they transfer part of that genetic material from one cell to the other.